If you took like a magnifying glass to the bottom of your foot and then immediately turned it to your scalp, you'd honestly assume you're looking at two completely different species.
Oh, absolutely. They don't look related at all.
Right. I mean, one bears the literal physical weight of your entire body against the ground, and the other grows this dense forest of hair while constantly fighting off solar radiation.
Yeah, the mechanical demands are just totally opposite.
Exactly. Yet, biologically, they rely on the exact same specialized building blocks to survive. We usually think of the skin as just this single uniform wrapper, just the wrapping paper keeping our internal organs safe.
Which is how it's been taught for decades, really.
Right. But today, we are going on a mission to completely decode the largest, most visible organ in the human body. We are going to look at the skin not as a static canvas, but as a complex, highly organized country, one with its own evolving geography, biochemical communication networks, and these really bustling cellular neighborhoods.
And the reason we can actually map this country in such unprecedented detail now is because of this massive, organ-wide spatial atlas of human skin. It was recently published in Nature Genetics, and it is our source material for today's deep dive.
And the scale of the source material is just staggering. I mean, we're talking about an analysis of 114 samples.
Yeah, taken from 15 different anatomical sites across 22 distinct human donors.
Which is wild. This isn't just looking at a tiny biopsy under a standard microscope. And that scale is what really hooked me because, well, the way we traditionally used to study tissue is basically like putting a city into a blender.
That's a really good way to put it, a biological blender.
Right. We would take a biopsy, grind up all the cells, run them through a sequencing machine, and just get a neat little spreadsheet of what was inside. You'd know the city had, say, 1,000 bricklayers, 50 bakers, and 10 mayors, but you had absolutely no idea where they lived.
Or who was living next door to the baker.
Exactly. Or who the mayor was actively yelling at across the street. You lose all the context.
Which is exactly why the technology used in this study is such a massive paradigm shift. Instead of a blender, they essentially used biological Google Earth. The technology is called MERFISH.
Okay, wait, MERFISH. What does that actually stand for?
It stands for multiplexed error-robust fluorescence in situ hybridization.
Okay, pause. That is a massive string of jargon.
I know, it's a mouthful.
Let's ground that for everyone listening. What does that actually look like for the cells in the lab? What's the actual mechanism?
So let's look at how it works. Normally, to read a cell's RNA, those are the transcripts, the chemical instructions telling the cell exactly what to do, you have to break the cell membrane apart.
Right, you have to destroy the house to read the mail inside.
Exactly, you destroy the architecture. But MERFISH completely bypasses that. It uses these tiny fluorescent glowing barcodes that bind to specific RNA messages right there in situ.
Meaning like directly in their original place inside the tissue. Yes, perfectly intact. So the researchers were able to capture the functional transcripts of 1.2 million individual cells, all while leaving the entire three-dimensional architecture of the skin totally undisturbed.
Wow. 1.2 million cells.
Yeah. And through this, they localized 45 uniquely distinct cell populations.
So we are seeing exactly who is standing next to whom and what they're actively doing. Let's look at the sheer granularity of those 45 populations because the study spotlights a couple of very specific cells to show off this resolution.
They do, yeah, to really highlight the power of the map.
There is one called SPNKC2, which is a specialized spinous keratinocyte, and then another called retic-fib2, which is a type of reticular fibroblast.
And their spatial locations are incredibly specific. So retic-fib2 is a highly specialized structural cell. The MERFISH data shows it diffusely spread deep through the dermis, but it's almost entirely restricted to the sole of the foot.
So it's a load-bearing cell, like the heavy-duty concrete foundation workers? Precisely. Yeah, but that first one you mentioned, the SPNKC2 keratinocyte, that one is found almost exclusively on the sole of the foot and the scalp.
Okay, this is the part that made me stop reading and just stare at the wall. The top of our head and the bottom of our feet. They are the absolute polar opposites of human anatomy.
Literally as far apart as you can get.
Why on earth would the body group these extremes and use the exact same specialized workforce for both?
It really comes down to evolutionary mechanics. Both the scalp and the sole are areas of immense, constant mechanical stress and friction.
Oh, I guess that makes sense. The sole obviously bears the kinetic impact of your entire body weight with every single step you take.
Right. But think about the scalp. It is constantly exposed to the elements, major temperature swings, and continuous mechanical friction from hair movement, brushing, grooming.
Huh. I never thought about hair movement as mechanical friction.
Oh, it definitely is. So the body evolved to utilize this specific SPNKC7 cell to build a highly durable, stress-resistant barrier in both of those extreme environments. It's a shared architectural solution to completely different environmental problems.
Man, that is fascinating. So let me take a step back and just trace the logic so far for our listeners, because I want to do a quick 5-minute recap here.
Go for it.
Bullet 1, we've spent decades blending up tissue to study it, losing all that structural context. But with this new MERFISH barcode technology, we've mapped the skin at a single cell level like a high-tech GPS, revealing 45 highly specific cell types.
Spot on.
Bullet 2, we are seeing that the body uses very specific cellular workforces for different mechanical jobs, adapting to extreme stress. So for you listening, think about why the skin on your heel feels so physically tough compared to the paper-thin skin on your eyelid.
Exactly.
They are both called skin, but they are built by completely different cellular construction crews. Have you ever really wondered how your body knows to deploy those different crews?
And that concept of different construction crews perfectly sets up the macrogeography of this atlas. Because the researchers didn't just want to know where 1 or 2 cells live.
Right. They wanted the big picture.
They wanted to understand how all 45 cell types were distributed across the entire landscape of the human body. And to map this, they utilized a mathematical algorithm known as the Shannon index.
The Shannon index. Okay, I know ecologists use that metric to measure biodiversity in a rainforest.
Yes, exactly.
They use it to calculate whether one patch of jungle has 50 different species of frogs and birds, while another patch down the river only has 2.
The mechanism is exactly the same here, but it's deployed as a proxy for cellular diversity in your tissue. And the spatial pattern that this algorithm uncovered operates on what they call a centrifugal axis.
Centrifugal, meaning like starting at a central point and spinning outward.
Precisely. If you look at the core of the human body, so the abdomen, the back, the buttocks, the Shannon index shows the lowest cellular diversity. The tissue architecture there is relatively simple and uniform.
Okay, so the core is basic.
But as you move outward along that centrifugal axis to the extremities, down the arms and legs, and up toward the head, the face, and the scalp, the cellular diversity just absolutely explodes.
Okay, so my central abdomen is essentially a quiet, rural farming town. The infrastructure is basic, it's steady and simple.
Right. Not a lot of different professions needed.
But my face, or my scalp, those are incredibly dense, highly diverse metropolitan cities. You have massive amounts of complex infrastructure packed into a tiny area. You've got hair follicles, sebaceous glands, pumping out oil, intricate sweat ducts. It is literally cellular Manhattan.
That is a perfect analogy. And the algorithm also picked up something highly counterintuitive about our joints.
Oh,
Yeah,
like
right.
the knees and elbows?
Yeah, specifically the flexural sites, meaning the inner folds of your joints, like the antecubital fossa on the inside of your elbow, or the space right behind your knee.
Okay.
Those areas are incredibly diverse. They actually show higher cellular complexity than the outer extensor parts of those exact same joints.
You know, actually, thinking about the physical mechanics of the body, that makes total sense. The inside of your elbow has to stretch, fold completely onto itself, manage intense sweating, and protect delicate superficial veins, all while being constantly in motion.
Right. There is a lot of biological engineering required for that to work without tearing.
But how does this abstract mathematical diversity translate to the physical realities of the skin, like how thick it actually is in certain spots?
Well, the study isolated a specific structural cell called the papillary fibroblast to answer that, and they call it papal fib for short. The spatial data proved that the sheer abundance of these papillary fibroblasts strongly dictates actual epidermal thickness.
So more of these fibroblasts equals thicker skin.
Yes. You find massive clusters of these fibroblasts in high-stress areas like the sole of the foot, the elbow, and the knee. They are the physical scaffolding workers that build up the thickness of the skin exactly where the body anticipates trauma.
I want to pause here because the picture we are painting is shifting so drastically from how we normally think about biology.
It really changes the whole paradigm.
It does. We are completely moving away from the idea of the skin as a generic wrapper. The MERFISH technology is showing us that skin diversity operates on a centrifugal axis simple at the core, wild and complex at the extremities. And specific building block cells, like those papillary fibroblasts, are actively dictating the physical thickness of your skin depending on what latitude and longitude of the body they are sitting on.
Exactly. It's highly engineered based on location.
But I mean, if we have this massive, sprawling city of 45 cell types, they can't just be wandering around aimlessly.
They aren't. Not at all. To figure out the zoning laws of this cellular country, the researchers deployed another spatial clustering algorithm. This one is called cell charter.
Okay, cell charter, what does that one do?
This algorithm analyzed millions of data points looking for proximity patterns. It was essentially asking mathematically, what is the probability that cell A is always found hanging out right next to cell B?
So it's literally looking for the social networks of the cells, who hangs out with who.
Right. And Cell Charter found a highly stable mathematical solution of exactly 10 multicellular neighborhoods that consistently appear across the entire human body.
10 neighborhoods everywhere.
Everywhere. These are distinct, highly ordered architectural zones. For example, there's N0DEJ at the dermal-epidermal junction, N-alveostroma for the deep structural tissue, N8 subcutis for the fat layers, and so on.
Okay, are we looking at all 10 today? Because that honestly sounds like reading a textbook.
No, we don't have time for all 10. We're going to zoom in on the most critical one for our immune defense. It's designated as N1 perivas I. Okay, break that down for me.
This is the superficial perivascular neighborhood. The spatial data revealed an incredible concentration of immune cells here. You have dendritic cells, T cells, and mast cells.
All the heavy hitters of the immune system.
Exactly. And they are all clustered incredibly tightly alongside specialized perivascular fibroblasts, specifically perivas fibi, wrapping directly around the superficial blood vessels just under the surface of the skin. Okay, I have an image for this. Think of this neighborhood like a medieval castle. The blood vessels are the main supply roads, bringing nutrients in and out of the kingdom.
I like this, yes.
So this N1 perivas neighborhood is your primary gatehouse. And the immune cells, the dendritic cells, T cells, and mast cells, they are your heavily armed guards.
Right, they're watching the roads.
They aren't just randomly patrolling the countryside hoping to stumble upon a threat. They are permanently garrisoned at the gates, clustered exactly where the roads enter the castle walls, ready to intercept any foreign invaders instantly.
That is perfect.
Yeah.
Because what you are describing visually is the physical manifestation of the biological concept called SALT. This stands for skin-associated lymphoid tissue.
SALT. I think I've heard of that in relation to the gut.
You're probably thinking of GALT for the gut. For years, SALT was understood conceptually. We knew the skin mounted its own localized immune responses, similar to how your tonsils protect your throat or Peyer's patches protect your gut.
Right, the local garrisons.
Exactly. But this study finally provides the exact spatial coordinates and the precise cellular roster of that localized immune garrison in the skin.
So for the listener, think about the last time you got a paper cut or like a minor splinter. Notice how the area around the cut swells up, gets red, and feels warm almost instantly.
It happens in minutes.
Right. That is not your body reacting from scratch and sending troops from your bone marrow all the way down to your finger. That immediate reaction happens because the immune guards were already stationed millimeters away in that N1 gatehouse, just waiting for the wall to be breached.
But here's the thing. Having a garrison stationed at the gatehouse is only the first step. The cells have to actively coordinate to stay there.
Oh, I see. They don't just magically glue themselves to the spot.
No, they don't. The structural fibroblasts building the skin and the immune cells defending it need a highly reliable way to communicate.
Because if the guards and the castle builders can't talk, the whole defense falls apart. So how are they communicating?
The researchers ran what's called a ligand receptor analysis to map out the biochemical phone lines between the cells in this neighborhood.
A ligand and receptor. Okay, let's translate that.
A ligand is essentially a chemical text message released by one cell, and the receptor is the antenna on another cell that receives that specific message.
Got it.
The core text message keeping this gatehouse functioning is a molecule called tumor necrosis factor, or TNF.
TNF. Okay, so who is sending the TNF text message and who's actually reading it?
The immune cells, the guards, are constantly broadcasting the TNF signal. And the signal is received by the perivascular fibroblasts, the castle builders. Specifically, it binds to a receptor on them called TNFR-SF1A.
Okay, so the guards text the builders. What happens when the builders read it?
When the fibroblasts read that TNF message, it triggers a critical feedback loop. The fibroblasts respond by lighting up a chemical beacon. They secrete a specific molecule called CCL19.
CCL19.
Yes, and that CCL19 acts as a homing signal, telling the immune cells exactly where to anchor and stay put.
Wow, it is a literal chemical codependency. Let me jump in and clarify this loop. The immune cells are texting the builders saying, hey, we are here, keep the lights on so we can see the gate.
Yep, that's the TNF.
And then the fibroblasts receive that TNF message and turn on the CCL19 chemical beacon that says, understood, guards, stay right here, this is your post.
That's exactly how it works.
So if you disrupt that TNF phone line, the beacon goes out, the guards lose their spatial anchor, and they just drift away, leaving the blood vessels totally unguarded.
Exactly. It represents a perfect homeostatic embrace between the body's immune system and the structural tissue.
Okay, we are about 10 minutes in, so let's do another quick recap and bullet summary for the listener.
An idea, there's a lot of biochemistry there.
Bullet 1, skin cells don't live randomly. They organize into 10 distinct architectural neighborhoods. Bullet 2, the N1 perivascular neighborhood is the skin's localized immune command center, the SALT.
Yeah.
And bullet 3, TNF acts as the crucial biochemical text message that keeps immune cells and fibroblasts locked in a protective embrace. So think about that. Your immune system isn't just floating around. It's actively texting your structural cells to keep the lights on.
It is a beautifully synchronized system.
Yeah.
But the reality of biology is that homeostasis doesn't last forever.
Nothing gold can stay. Right.
Exactly. The atlas didn't just map a single moment in time for young, healthy skin. It also captured the time dimension.
Oh, meaning how the skin ages over time.
Yes. They analyzed how these neighborhoods structurally degrade as we age, specifically looking at older abdominal tissue.
Okay, so we move from how the city is flawlessly built to how it inevitably decays. What is the physical mechanism of aging in these cellular neighborhoods?
It is basically a massive demographic shift. The deep structural scaffolding of the skin is governed by a neighborhood we mentioned earlier called N4 stroma.
Right, the foundation workers.
As we age, this N4 stroma neighborhood physically shrinks. The MERFISH data shows this is driven primarily by a sheer numeric loss of retic fib eye cells.
Retic fib eye. Wait, we talked about retic fib dust being on the sole of the foot earlier.
Exactly. Retic fib fust is another structural variant. They are the primary workers responsible for producing dense collagen throughout the body.
So the primary workforce literally clocks out, retires, and just disappears.
They really do. And as that stroma neighborhood shrinks and the collagen drops, another neighborhood takes over the vacant space. The N3 perivascular 2 neighborhood, which houses deeper blood vessels, smooth muscle cells, and high endothelial venules, begins to relatively expand.
Wait, let me push back on this. If the vascular neighborhood, the blood vessels, is expanding and taking up more space as I get older, shouldn't older skin have better blood flow?
You would think so, right?
Yeah, I mean, shouldn't it be healthier if it has a more prominent vascular network? Why does my grandmother's skin look almost translucent and bruised from the absolute slightest bump if she has more of this vascular neighborhood?
It's a great question, but you have to look at the structural context, not just the raw volume. The vascular neighborhood isn't expanding because it is growing new, strong, healthy blood vessels.
Oh, okay.
It is expanding relatively because the thick collagen scaffolding, the retic fib eyes that used to compress and protect it, has completely vanished. The structural integrity is just gone.
I see.
Those deeper vessels become physically more prominent and closer to the surface, but they are incredibly fragile because the bouncy protective padding that used to surround them is missing.
Ah, I get it now. It's like the thick foam insulation padding around a water pipe in your basement rotting away over the years.
Yes, great visual.
The PrEP suddenly looks bigger and more exposed to the naked eye, but it's actually incredibly vulnerable to bursting because the protective sleeve is just completely gone.
Exactly. So aging isn't just some generic, abstract loss of elasticity like lotion commercials tell you. It is a highly specific demographic collapse in your cellular neighborhoods. The scaffolding workers abandon their posts, and the vascular structures are left entirely exposed to the elements.
Wow. Have you ever noticed how an older relative's skin can tear or bruise from the slightest bump? Well, now you know the exact cellular workforce that went missing to cause that.
It's sobering, but it's the biological reality.
I want to weave all these threads together for a second because the narrative is getting incredibly complex, but so cool. We've established that the skin is highly organized into stable architectural zones, like the N1 immune gatehouse.
Right.
We know these cells use chemical text messages like TNF to stay locked together, and we know that the mere passage of time causes the structural workers to vanish, leaving the deeper networks vulnerable.
That's the baseline.
But aging is a slow, predictable process. What happens when these tightly controlled neighborhoods face a sudden catastrophic breakdown, like a disease?
That is exactly what the researchers wanted to know next. To investigate active disease states, they utilized a completely different spatial technology called Visium.
Visium, how is that different from MERFISH?
Where MERFISH is incredible at identifying single cells, Visium is designed to map slightly larger spatial microenvironments across entire cross-sections of diseased tissue. They used it to map the architecture of 5 severe clinical conditions.
Okay, which ones?
Atopic dermatitis, psoriasis, hidradenitis suppurativa, basal cell carcinoma, and squamous cell carcinoma.
That is a wildly diverse list of diseases. I mean, you have severe eczema, psoriasis, incredibly painful skin tunneling, and 2 entirely different types of skin cancer.
There was a lot of ground, yeah.
Did the spatial algorithm actually find any common ground between an autoimmune disorder and a spreading tumor?
It did, and it fundamentally changes how we view skin trauma. The Visium data uncovered a universal stress neighborhood on the surface of the skin. Yes. Regardless of whether the tissue was under attack from a multiplying cancer like squamous cell carcinoma or tearing itself apart through severe autoimmune inflammation like atopic dermatitis. The keratinocyte cells on the surface exhibited a universal, uniform stress response.
Wait, really?
Yeah, they all ramped up the exact same distress genes, specifically ones called KRT6A and S100A8.
Let me process that. Are you saying the surface skin cells reacting to cancer look exactly the same as the cells reacting to eczema?
Well, not the cancer cells themselves, but the surrounding civilian skin cells reacting to the crisis. They throw up the exact same molecular flags regardless of the underlying cause.
Oh, wow. It is like a peaceful city where a massive riot suddenly breaks out. The civilian buildings just start hanging universal distress flags out the windows.
Right, they just know there's a crisis.
It absolutely does not matter if the city is being invaded by a foreign army, which would be the cancer, or if it is a civil war tearing the city apart from the inside like an autoimmune disease. The visual stress flag on the surface looks exactly the same to the outside observer.
That's exactly it. But the most profound and frankly terrifying discovery happened back at our N1 perivascular gatehouse.
The immune garrison, what happened to them?
In almost all of these severe diseases, that localized immune neighborhood gets completely hijacked. It stops functioning as a peaceful border patrol and drastically expands its footprint. It becomes this massive pan-disease immunomodulatory hub.
So the guards panic.
They completely panic. They start frantically pumping out chemokines and interleukins, essentially sounding chemical air raid sirens, which calls in massive, overwhelming amounts of systemic inflammation from the rest of the body.
So the gatehouse turns into a chaotic, expanding military zone that just destroys the surrounding city.
Sadly, yes. And the study highlighted a very specific clinical case to show just how extreme this architectural remodeling can get. They looked deeply into the spatial mapping of hidradenitis suppurativa, or HS.
HS, that is a devastating chronic inflammatory disease.
It really is. And the Visium data isolated a unique spatial signature in HS. They labeled it the C14 cluster.
The C14 cluster, what does that do?
This specific cellular cluster physically corresponds to the formation of epithelial tunnels deep beneath the surface of the skin.
Tunnels, like literal physical holes.
Yes. These tunnels are the defining hallmark of advanced HS, causing chronic infections, immense pain, and severe scarring. The researchers were able to visually map how the total breakdown of the normal stroma, combined with that violent hijacking of the immune gatehouse we just talked about, literally carves these pathological tunnels right through the human tissue.
Wow. Just, wow. The mental image of the body's own immune garrison expanding and physically carving tunnels through the skin is terrifying.
It's intense, right?
It really is.
Yeah.
But at the same time, it is incredibly empowering from a medical standpoint. For the first time, we aren't just guessing why the skin is collapsing. We have the exact architectural blueprints of the destruction.
Exactly. We can see the rioters on the map.
Okay, 15-minute recap here. Bullet 1, aging dismantles the structural stroma neighborhood, leaving vessels exposed. Bullet 2, completely diverse diseases trigger a universal stress response in the skin's surface civilian cells. And bullet 3, That perivascular immune neighborhood gets hijacked and drastically expands to fight off disease, physically altering the skin's landscape, like carving those tunnels in HS.
Perfect summary. And that leads directly to the ultimate takeaway of this massive atlas. The implications for the future of medicine here are truly staggering.
How so?
Because we now know the exact latitude and longitude of these specific cell populations, the entire field of regenerative therapy can pivot. Imagine a future where, instead of applying a generic broad-spectrum anti-aging cream to the surface, a dermatologist could inject specific, lab-grown SPNKC2 cells or soul fibroblasts directly into thinning areas of the skin.
Oh man, you could biologically rebuild the missing scaffolding from the ground up.
Exactly. Or think about chronic autoimmune diseases like psoriasis or HS. Right now, patients are often prescribed systemic biologic drugs that suppress the entire body's immune system just to clear their skin.
Which comes with a lot of side effects.
Right. But if we know the exact biochemical phone line, that specific TNF connection happening right at the N1 gatehouse, we could design therapies that target that communication line spatially.
So we could cut the phone line and calm the riot down at its exact spatial route without compromising the immune system in the rest of the body.
The level of precision we are moving toward is unprecedented.
Yeah.
We are finally transitioning from treating the skin as a single flat piece of canvas to treating it as a dynamic, multi-layered ecosystem.
Which leaves me with a final provocative thought for you to mull over as we wrap up today's deep dive. If we now have the hard data proving that your skin's cellular makeup, its physical thickness, and its localized immune set points change drastically depending on whether you are looking at the inside of your elbow or the center of your abdomen.
It really makes you rethink your medicine cabinet.
Exactly. Should the way we apply medicine or even everyday skincare be radically different depending on the exact geography of our body? If your face operates like a highly complex metropolitan city and your back operates like a simple rural town, why are we treating them with the exact same broad-spectrum lotion?
That's a great question. It really makes you wonder what the skincare aisle of 2035 is going to look like.
It's going to be wild, that's for sure. Thank you for joining us on this deep dive into the source material. We hope you think entirely differently the next time you look in the mirror. You aren't just looking at a simple wrapper. You are looking at a brilliantly engineered, incredibly diverse cellular country. See you next time.